This is a recording of the Chapter 44 Osmoregulation and Excretion lecture. We're going to be talking about peeing all lecture, so I recommend you pause right now, go to the bathroom, and then come right back. And don't drink a big cup of water or coffee because you are going to have psychosomatic suggestions the entire time. All right, so you know how basic permeability and diffusion work. If you don't remember how these work, you need to go back in your textbook. Chapter 7, which talks about diffusion and osmosis and things like that. Generally, always remember that things try to move from their higher concentration to their lower concentration. And in the case of something that is hypotonic, the water will move from the higher concentration of water towards the hypertonic thing, which has less water in it relative to the amount of solute. So if you have a 1% salt solution next to a 0% salt solution, that is the same as a 99% water solution next to a 100% water solution. And that's the way I want you to think about it. Also note that they speak of osmolarity instead of necessarily hypertonic and hypotonic. For the purposes of this course, there is no real difference between hyperosmotic and hypertonic, other than the fact that hyperosmotic is assuming to begin with that there is going to be a barrier between the two, so that you are going to have a membrane and this will be osmosis. Technically, hypertonic, for example, doesn't assume the presence of a membrane, so it could be simple diffusion going on. Now, some basics to remember about aquatic life forms is that they are surrounded by water. So they have two things that they have to consider simultaneously. One is water, and two is salt. So what fish need to do is if they're freshwater, is they need to get rid of extra water. But what they need to do is concentrate their salt. So they will have pumps on their gills that will actually pump salts and solutes in general out of the water and into their bloodstream. And then these fish will pee constantly. They'll excrete high volumes of very dilute urine because they are not taking extra special efforts to keep their water. They're taking extra efforts to dump their water. Whereas in a freshwater, saltwater, the reverse is going to be true. You're going to have gills that pump out the salt, and you are going to have urine that is extremely concentrated because saltwater fish needs to keep the water in its body, but needs to dump salt like crazy because it's constantly taking it in through body surfaces, and also every time it drinks or eats something. All right, so there are some fish that actually have to spend part of their life in freshwater and part in saltwater, and so they have to reverse these profiles as they make their changes. Now, when considering osmoregularity, remember this is both about salt and water, and depending on who you are and what you do for a living, you can have various inputs and outputs of water. So, for example, if you're a kangaroo rat that practically lives in the desert, some of your food is going to be wet, but the majority of the water that you gain is going to actually be involved in metabolic water. So remember, when you convert sugar into carbon dioxide gas and water, these things burn energy not only to stay warm and stay alive, but to specifically create the water they need to survive. Similarly, the water loss profiles are going to be different. So a human over here who lives in, usually environments where they can acquire water. Humans did not evolve in dry places. We evolved in jungles and savannas where there was water to be had. We're going to drink more of our water and make less of it out of creating it, but we will have a higher ingested value because our food is going to be wetter. If you flip around, you'll look and see that humans pee out a lot more water than a mouse does. Okay, the kangaroo mouse spends a lot of time trying to keep that water out of its urine. Now, throughout this chapter, we're going to be dealing with the concept of transport epithelia. The gills that I just talked about, where they're pumping in salt, those count as a transport epithelia. Epithelial tissue is a barrier, and transport epithelium specifically has goals of transport across it. We've actually already talked about these when we were talking about the small intestine. It is an example of transport epithelia because it's trying to transport food molecules from the lumen of the small intestine into the bloodstream. So there you go, that's a transport epithelium. But also when we talk about any kind of excretory tissue, the nephridia, when we talk about the nephrons, when we talk about anything involving the kidney, we're going to be talking about transport epithelium and the concept of a differentially permeable, not just membrane, Because that's the other problem. Differential permeability to you as a plasma membrane thing. This now, transport epithelia, is about multiple cells, all with their plasma membranes forming a kind of a barrier of some sort, and how they differentially move things back and forth. So what was happening at a one-cell stage is now going to be happening at a multi-cell stage. So when we're talking about osmoregulation and excretion, we're talking about maintaining water balance, we're talking about maintaining salt balance, and also we're talking about waste balance. Here's the short and sweet of it. Anytime you metabolize proteins and break them down, you're going to create some ammonia, okay, because of those amino groups are going to get given off as a waste. Ammonia is highly toxic. Drinking it is lethal. So we've got to do something about the ammonia, and you have different options. If you're something that lives in water, even if it's fresh or salt, now notice this isn't about acquiring water. This is now about relieving yourself of waste. If you're something that lives in water, most of the time you don't even convert the ammonia. You just dump it out of your body directly. Fish, part of the reason they smell fishy is because their body tissues just secrete the ammonia directly into their surroundings. It's found in their muscle tissue, it's found on their skin, and it's why fish are both basic, okay, because ammonia is a base, and why they taste better when you put lemon juice on there, because it neutralizes the ammonia. Okay, so very strong smelling or strong tasting fishes, people will squirt lemon juice on them and it moderates the taste, because they keep the ammonia as is. Now, a lot of organisms instead convert that to urea. It is much less toxic than ammonia. Problem, though, it takes energy to do that. And then you have to get rid of it still. Now, why doesn't the fish do this? The fish can pee constantly throughout its time. It can just let the ammonia diffuse into the surrounding waters. It doesn't have to work hard to get rid of ammonia. But if you just started sweating ammonia, it would stick to your body because you're not bathed in water. So also it takes a little bit of effort for you to find a place to pee. Okay, you can't constantly just be leaking urine all over yourself because you're not swimming through water. So you do that urea trick. However, your life is spent around the idea that water is plentiful and reasonably easy to find. Birds and other reptiles, which didn't necessarily evolve in as wet of conditions, do something called uric acid conversion. It takes even more energy than making urea, but in the process what they do is they made a solid. Urea is a liquid. Uric acid, when they strip it out of the bloodstream, turns into a particulate matter, and it's very easy to specifically isolate uric acid. It's easy to pump. When we get into kidneys and the complex arrangement of things in the kidney and all the tricks that we're going to do, the whole purpose of the kidney is centered around this one fact. You don't have a urea pump. You have no way of specifically pumping urea. It's actually fairly difficult to do. So all of the kidney is going to be based around of squeezing the fluids of the blood into a space and then taking everything back except the urea. That's the basic trick. So you have to work very hard because you don't have a simple urea pump. Well, reptiles and some insects have uric acid instead as their product, and uric acid pumps are plentiful, so they can just dump it directly and put it with their feces, and that's why birds don't pee and poop. They excrete both at the same time. The uric acid is the white stuff. Okay. We've already talked about this stuff. The key to urea being is that it can circulate through your bloodstream without killing you because it's much less toxic. It's like 1/10,000th the toxicity concentration. So, or I should say it can be 10,000 times more concentrated before reaching toxicity. All right, when you're excreting, you do several steps. The first one is most, probably the one that is most misunderstood, if we're going to be honest. It's the filtration step. It is where you take the bloodstream and you squeeze it through a capillary bed into some kind of catch vessel. And so what's going to be squeezed out is basically the plasma of the blood, or honestly whatever else is in interstitial fluid. So every time, just as in the previous chapter, your heart was pressing liquid out of the bloodstream and it was flowing into the interstitial fluid, and we had sugars there and salts and some proteins, but more oxygen dissolved gases. That's the same kind of stuff that will be squeezed out at the beginning stages of trying to excrete problems, or trying to excrete your waste. So that's the filtering step. It's where you squeeze it out through the capillary beds and you retain the biggest stuff, okay, simply based on size. Then there's a reabsorption phase where you try to pull back in, for example, the water. Because if to filter your blood, you have to squeeze the portion of your blood that you want to get rid of the urea from. Let's say you isolate a specific chunk of blood. all of the fluid volume of that blood has to go into the catch basin. It has to all be squeezed out. And so therefore, the entire fluid volume of your blood would get dumped in the toilet. Because if we filter all of your blood and we never reclaimed the water, all of the water in your blood would end up in the toilet, and you would have to drink your entire blood volume each day in water. You'd also lose all the salts that were in the blood. You'd lose all the sugar that was in that blood. So every time you could, a great diet mechanism in that case would be just simply drinking a lot of water so that you had to pee more, so more sugar left your body. So we've got to reabsorb. Finally, we got to secrete some stuff back into the bloodstream, and it loves to say secretion of wastes, but remember, you don't have a urea pump, so you don't pump urea, the key waste. So I don't really like saying secretion of waste. I just like to say you're putting stuff back into the filtrate you want to get rid of. They're not specifically waste. And then excretion is basically when you declare you're done with the process. It's as concentrated as it's going to get, and you dump it into the final catch basin. And in my class, that's the bladder. So you can't hold your pee in, you can't hold your urine in longer in hopes of reclaiming water. That doesn't help you. If you think you're dehydrated, don't hold it in. That's not going to help you at all, because once the urine has gone to the bladder, nothing gets reabsorbed at that point. We're done with reabsorption phase. In the bladder, that's excretion. It's only a matter of time and preference when it finally leaves the body. At this point, it's already gone, from the opinion of a biologist. Okay, and this is just a pictorial version of that. We'll get into a much more complex arrangement of that when we talk about the kidney. Now we are going to move into the concept of a renal cortex versus renal medulla. And cortex and medulla are going to be used several times. They're going to be used in the brain. They're going to be used in adrenal glands. They're going to be used in the kidney. So let's get down to it. A cortex is an outer layer, and a medulla is an inner layer. That's going to be important today because you'll need to know which parts of the nephron, the portion of the kidney we're going to be working with, are in the cortex, which will be less salty, and which are in the medulla, which is going to be increasingly salty as we dig deeper and deeper into it. I'm going to leave you to understand the renal artery, which is coming away from the heart, and the renal vein, which is traveling towards the heart. And I'm also going to leave it to you to just basically understand that the kidney goes to a ureter, which goes to the urinary bladder, which goes to a urethra. And that doesn't matter if you're male or female. That's what's going to happen. Now, the basic nephron organization is this. We're going to have a capsule, a catch basin. And inside, it's going to be the Bowman's capsule. Inside is a capillary bed called the glomerulus. That's where your filtration step happens. Everything that goes into the Bowman's capsule, if it doesn't get reabsorbed during the reabsorption phase, it's going to end up in the toilet. So when you filter, everything that goes out into the Bowman's capsule has to be considered on its way to the toilet. This is backwards from the way most people think about it. So focus very, very hard on remembering that if it gets filtered into the Bowman's capsule, if it gets squeezed through the capillaries and pushed in, it's in the toilet unless you do other work. Then we're going to do a proximal tube where we're going to do some secretion and reabsorption. We're going to have a descending limb and ascending limb of the loop of Henle. We're going to have a distal tube, which we're going to do some of the same tricks that we did in the proximal tube, just doing them again. And then we're going to have a collecting duct, which is headed towards what's called the renal pelvis, which heads towards the ureter, which heads towards the bladder. So, functionally speaking, once a liquid has reached the renal pelvis, right here, this area, it's as good as in the bladder. It's just a matter of time, and anything in the bladder is as good as in the toilet. So once you hit this spot, that's when you're going into the excretion phase. The final phase is because now it's in here. So don't think of excretion as going to the bathroom. Think of excretion as the thing that makes you need to go to the bathroom. It's already done by the time you feel the urge to pee, because that stuff isn't coming back to your body. There's no return. It's already gone. So now, I am skipping to the part where you are already in the Bowman's capsule because you've been squeezed through the glomerulus. This step is just relying on blood pressure, just as before, just as liquid is squeezed out of any capillary. Once you are into the ducts here, once you're in this yellow tube, you are headed towards the toilet. So if we want to keep anything of you, we've got to do some work. The way we're going to do that is with transport epithelia and that transport epithelia is going to be playing permeability game. In some locations, it's going to be pulling liquid out. How's it going to be doing that? Well, one example is it will pump salt out of the proximal tube. And as the salt concentrations go down in the filtrate being the liquid in the tube that's headed towards the toilet, as salt concentrations go down, suddenly it treats the interstitial fluid surrounding the kidney and inside the kidney as hypertonic, right? Because now it's lower salt. It was just blood. It was the tonicity of blood, 300 millimolar salt, for example. So if the kidney pulls some of the salt out of that filtrate, and now it's only 200 millimolar salt, now it's higher water concentration. That water is going to try to fly out into this interstitial space and into the interstitial fluid. And just as things that are in this ductwork are headed towards the toilet, if you pull it out and into the body of the kidney, now it is being retained by the body. It is going to go back into the bloodstream, and it's been saved. So anything left in the ducts is going in the toilet, and anything pulled out of the ducts and into this area out here, or into the medulla, or any of the portions, that's the stuff you have saved. This is the second place that is very difficult to remember. The first difficulty was remembering that fluid in the duct is headed towards the toilet. The second thing is that anything pulled out of the duct and into the tissues of the kidney is headed back into the body. It feels like when you say it goes into a kidney, you want to think it goes into the bladder somehow, but it doesn't. If it goes into the body of the kidney, it is staying in the body. So then that's an example of how you can pump out solutes on one side and cause water to follow. In the descending loop of Henle, what we're going to do is we're going to have a loop that goes down, down, down from 300 millimolar salt to 1200 millimolar salt. Okay? As we do that, we simply allow the ductwork to be permeable to water, but not to salt. Then, as you switch around and start heading back upward, we switch the permeabilities. Now it's permeable to salt, but not water. And then even we'll pump some salt out, but still not allow water out. This is a countercurrent exchanger. And let's zoom in on it. Here's what it really looks like. As your 300 millimolar solution, or milliosmolar I should say, comes downward, it's encountering an increasing gradient of salt. So the least concentrated stuff for salt encounters a slightly more concentrated area and loses water to it, thus becoming more concentrated. Salt can't leave, but water can, so we're shrinking volume here. As we get down deeper into the medulla, the fluid is encountering an even saltier environment, or an even higher osmolarity environment. So more water is pulled out until you reach equilibrium again, and we've concentrated this. Now this stuff is twice as concentrated, which means it's half the volume it used to be. We've pulled half the water out. We go all the way down to the bottom, and what we have is we have a fourth of the volume all of a sudden. So a fourth of the liquid is remaining because the rest was pulled back into the kidney. Wow, that was cool. Now, as we go back upwards, we switch the permeabilities and allow salt to leak out, but not water. And so as you have a 1200 millimolar salt or 1200 milliosmolar concentrated solution come upward and visit something that's less concentrated, 900 here now, it'll let some of the salt leak out until it can reach equilibrium. And as we get further up, it will deliberately pump the salt out now to where it gets even less concentrated, to where now it's even less concentrated than the cortex. When it gets up to the top of the cortex here, let's go backwards and see what happens, because it's more like water than this stuff out here is. This stuff out here is 300 milliosmol. See? This stuff is 100 milliosmol. If we open up water channels again, if we let it become permeable again, water is going to leak out into the interstitial fluid, and we're going to concentrate our solution even more. And so look, that actually happens. Because of the salt pumping out, because there's a third as much salt or osmolarity in this tube as there is in the surrounding tissues, more water comes out. So if you're doing the math, we concentrated it by a fold of 4. Okay, so it's a fourth the volume. Then we got rid of 3/4 of our salt as well. And then we continued to get rid of the salt until up here we had 1/12 of the original salt. Because we started down here with something that was of a fixed volume at 1200 millimolars, and then we sucked it all out until it was 100 millimolar. So now we've recovered the vast majority of our salt. Open up some water channels, and it concentrates to a third of the current volume it is up here. Remember, over here it was like a quarter of its volume or less because we'd already pumped some water out. Okay. Then now we cut it down to a third of the quarter, so that's 1/12 of its volume. And then we're going to come back to the collecting duct, and we're going to play the exact same trick again. We're going to have a membrane that is permeable to water, but not salt, okay, allowing this thing, as it goes down, to become more concentrated for salt and water to be reclaimed. And if we get it all the way down to the 1200 mark again, our 12 times concentrated stuff, or maybe even 20 times, taking into account what happened up here, can now be at the point of, hey, that's pretty crazy. It's 4 times more concentrated again. So we're between 48 and 80 in fold concentration. Okay, so we have reduced its volume extraordinarily small. So it's 1/50th of its original volume of liquid. Did you follow that? That was the game. Now, what was the whole purpose of this? We left urea inside this liquid for most of the time. Problem. You need to jumpstart the kidney. It's a chicken and egg problem. How does the kidney get salty to begin with? Well, one of the things you do is allow urea to leak out of the collecting duct just a little bit, and that sets up some of the osmolarity gradient in the intermedulla to begin with. Some of the rest of it is maintained by sodium being pumped out at higher levels, but down here it's that leaking of the urea that sets up that concentration of the intermedulla. Because of that, a lot of the urea leaks back in here, so you even supercharge it with urea. It's kind of a chicken and egg there as well. But that's the purpose of letting some of the urea leak out, is to set up that osmolarity gradient down bottom to begin with. Okay, so did you follow that countercurrent game? As it was going down to saltier levels, we allowed water to move. So that it was always getting saltier. As you came up through less salty levels, we allowed salt to move, so it was getting less salty as it came. And then we flipped back down and did the exact same thing as we did in the descending loop. This is the key to how the kidney works in us mammals and how it's so efficient. Note, reptiles don't need this because they are not going to secrete their salts and their urea waste through a kidney. They're going to secrete it through their intestinal tract. They're simply going to pump that uric acid from the bloodstream into the intestines and let it go out as poop. So, in mammals, this thing called the juxtamedular nephron, okay, is this idea. A nephron that goes deep into the medulla and comes back out, allowing this salt concentration. In mammals that don't focus as much on salt, okay, and maintaining their water, you have shorter or non-existent juxtamedullary nephrons. Okay, so you wouldn't expect to see them as much in a freshwater mammal like a river otter, because they can drink all they want. They don't need to worry about water concentration as much. So you tend to have shorter loops of Henle in these guys. Okay, reptiles, once again, don't have these complicated loops of Henle. They don't have these juxtamedular nephrons. Anytime they do have a kidney, they do have it. They use cortical nephrons further closer to the surface of the kidney that don't have this loop of Henle in them. All right, now, just skipping along, how do we regulate the amount of water being dumped out of the body? Here's a little bit of hormone regulation. Antidiuretic hormone. Okay, diuresis means peeing. So an antidiuretic hormone makes you pee less. So that means you keep the water. The kidneys are always operating. They never stop. So if you're going to pee less, that means you still want to excrete your ammonia waste, your urea, but you want to do it in a smaller volume. This is the third place where it is very difficult to keep track of things. Antidiuretic hormone makes the collecting duct more permeable to water. What will that do? That might surprise you, because you're like, if it makes it more permeable to water, doesn't water leak out? Yes, that's the point. Remember, if you're in the tube, you're headed towards the bladder. If we make this thing very water permeable, water is allowed to leak out, thus concentrating the urine. If we take away antidiuretic hormone, then now it's not there. A lot of these water channels close up, and now instead of concentrating so much as it goes down the collecting duct, it stays diluted, which means more water goes into the toilet, which means you are not conserving your water as well. So, antidiuretic hormone If you follow that, when it kicks in, channels for water open in the collecting duct, allowing water to rush out of the collecting duct and into the body of the kidney. And remember, anything that goes into the body of the kidney, the interstitial fluid, is reclaimed. Okay, so that's being reclaimed, that's being kept. Anything that stays in the collecting duct goes to the toilet. Vampire bats, when they feed off of the blood of a cow or something, they shut down their antidiuretic hormone entirely. They want to shut these. They want to shut all of these water channels because they don't want to conserve water. They're drinking large amounts of water. Blood is a very dilute food source. So what they do is they want to pee like the dickens. Plus, because if they drink a lot of this water, they can't fly. Okay, now they really need to pee. So this is kind of your original insult to injury scenario. The bats first drink your blood, then they pee all over the cow before they can take off again, because they have to get rid of that water weight before they're light enough to fly. So if you want to catch a vampire bat, catch it in the early stages of a feeding cycle. It's gained too much liquid to fly, but it hasn't had to pee yet, so it can't get anywhere. Okay, that was antidiuretic hormone. You should diagram that. You should draw yourself out a pattern of arrows or a concept map of how that affects the body's ability to conserve water. In summary, high antidiuretic hormone, high ADH, means low pee volume, means water conserved. Incidentally, alcohol interferes with antidiuretic hormone. Okay, since antidiuretic hormone makes you pee less, interfering with it with alcohol would make you pee more. And that's one of the reasons why you pee a lot more and you have to go to the bathroom more when you drink alcohol, far out of proportion to the volume you're drinking. It's not just that you're drinking lots of liquids, it's that you're specifically dumping the liquids out of your body. That's also part of the cause for dehydration when you're drinking lots of alcohol. It's because your body isn't working as hard to maintain the water volume. It's not about drinking the alcohol and the alcohol somehow dehydrates you. It's about the alcohol interfering with ADH. And that's what starts to dehydrate you faster than anything. That and destroying the alcohol also takes up water. So your liver burns up water as it gets rid of the toxic alcohol. Here's what conceptually it looks like. The pituitary gland, which we will study ad nauseum later when we do hormones and when we do the nervous system, it has osmolarity receptors in it, so we're sensing the osmolarity of the interstitial fluid once again. And just as with pH, salt concentrations can denature a protein, so it's very easy to make a sensor that changes shape when the blood gets too salty or not salty enough. Oh, did you pay attention to that? This is based on saltiness. This is not based on water volume. So this thing is not measuring, for example, the volume of blood you have in your body. It's measuring the saltiness of the blood. So if your blood gets too salty, the ADH kicks in and says, whoa, 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 the blood is too salty. If we pee in a large volume, that won't help us get rid of extra salt, but it will dump lots of water, and that'll make us even saltier. That's a problem. We're getting too salty up in here. So what we need to do is conserve our water. So see, just like in respiration, where you weren't sensing the oxygen levels in the blood, you were actually sensing the pH, which allowed you to infer that there was too much carbon dioxide, which allows you to infer that there's not enough oxygen. Well, here, the higher osmolarity infers that you're low in water, because otherwise, why would your blood be so salty? Noting this, if you eat lots of potato chips, it can cause you to retain water, because if you're eating lots of a salty substance, ADH kicks in and says, whoa, whoa, we need to maintain our water volume or else the blood is going to get too salty and we're going to have problems. Aquaporins from chapter 7 are that specific channel that help water move across plasma membranes. Remember that water can't move at full speed. It can at maximum move a third of its speed without help going through a plasma membrane, because plasma membranes are nonpolar and water is polar, so that does slow it down. Aquaporins are the little holes that you create that allow the water to diffuse much more rapidly. And this is the channel that is both opened and created when ADH hits the collecting duct. Oh, hey, there's a picture of it. Hey, do you remember cyclic AMP? Remember that second messenger? Cyclic AMP? You really should go back and look at chapter 7 and the signaling and the water going in and out and all that stuff. It's really about this. Heck, there's even some exocytosis and vesicles here. where these vesicles that already have aquaporins on them are fused with the plasma membrane, allowing water to come through them. So really saying that they create the aquaporins as needed is a little bit of a misnomer because it would take too long. Instead, they already have them on board and they push them onto the plasma membrane. And similarly, when you run out of ADH, these things get endocytosed back into a vesicle. Now, we were talking about the saltiness of the blood. That's one way to measure the problem, but what if the blood wasn't salty? What if the blood was leaking out of your body? See, ADH would help you if you've eaten too many salty things and need to get more water in you, or retain the water that is already in you, lest your blood salt levels go up even further as it concentrates because you're peeing. But what if you've been stabbed and you're bleeding? Okay. And you're not like lethal bleeding, but you're losing a reasonable amount of blood. You need to keep water in your body once again because you need to keep that blood volume high. Okay, or what if you've been dehydrated for a long enough period of time, or you're sweating a lot? Hey, how about that one? When you sweat, you lose both salt and water at the same time because your sweat glands cannot reclaim the salt. So they just excrete liquid that is, the saltiness of your blood. You're losing salt that way. So if you sweat a lot, that level of dehydration wouldn't register with ADH because the overall saltiness of the blood is the same. You lost the salt and the water at the same time. But you still need to retain water because you're running out of water. Even though your blood is the right saltiness, you're about to die of dehydration. So how do we sense things like that? Well, then there's the RAS pathway, the renin, angiotensin, aldosterone system. Okay, so the glomerulus, that network of capillaries, you have a sensor. Okay, you have a sensor in that region, and the arterioles going into that senses the blood pressure coming in. Why is it there? For no other reason than the kidney is part of this game to begin with, so you might as well put the sensor at the kidney. If that sensor sees a drop in blood pressure, that means that blood volume has gone down, either because you bled or because you sweated out too much of the salts and water. Okay, so regardless of how salty the blood is, we don't have enough of it because the blood pressure is too low. The glomerulus at that point will be releasing some enzyme called renin. It's actually the juxtaglomerular apparatus. And that enzyme, renin, is going to start a cleavage pathway. Okay, so there's your juxtaglomerular apparatus. It's going to be sensing the blood pressure. Okay, and what you're going to do is it releases renin. It converts angiotensinogen, I should say, which is in the bloodstream, into angiotensin. Okay, and then you get conversions into angiotensin 2, yada yada. What does that do? That causes the arterioles, first off, to restrict artificially. They constrict. That drives up the blood pressure. Okay, because we got to keep the heart beating, because maybe we're bleeding out, and a critical loss in blood pressure could cause the heart to, go heart attack on us, could go into fibrillation if it tries to suck on a vacuum. Okay, so that's one thing. Second of all, it tags the adrenal gland. The adrenal gland releases the aldosterone. So there's our renin, angiotensins, aldosterone pathway. Aldosterone causes the distal tubes to reabsorb more water and more salt, both. Okay, let's look back at it. Here we go, back up to the picture. Okay, distal tube. Remember, the distal tube pumps out salts and allows water to follow? Well, if your blood volume is getting too low, we don't want to specifically get water but not salt. We want more of everything, because if you've sweated it out or bled it out, we need both the water and the salt. So what happens is the aldosterone, okay, comes over here and tells this region to, okay, pump out more salt, hyperactivate these pumps, and allow even more water to flow after it. Okay, that's going to reclaim blood volume. So let's get back down, sorry for the flashing, to what's going on in the RAS pathway. This is another way that you keep your blood pressure, your blood volumes high, and your water volumes plus the salts. So the first way, ADH, was measuring salt concentrations but not concerned with volume. This way, the RAS pathway was measuring volume but not concentration. The two cooperating together help maintain your salt and your water amounts in your blood approximately correct, and also the approximate correct volume of blood in your body to begin with. Okay. There is an opposing hormone that opposes RAS, atrial natriuretic protein. Okay, natriuretic protein. Good Lord, these ones are hard to say. Atrial natriuretic protein. Uresis is peeing. Okay, atrial natriuretic protein is going to cause you to pee more. All right, so if you have an increase in blood volume that's too high, a pressure that's too high, this thing is going to inhibit the release of renin to begin with. And you've got different locations where this sucker can pay attention to. All right, so not only is this trying to slow down the RAS, it's actually preventing the release of the renin in case something's malfunctioned at another location. Because maybe your kidneys are having high pressure, but other parts in your body or low pressure, but other parts in your body are actually high pressured. So we got to have a backup to the backup. And that's the end of this lecture.